Flowers are the seat of sexual reproduction in angiosperms. In the flower, androecium consisting of stamens represents the male reproductive organs and gynoecium consisting of pistils represents the female reproductive organs. A typical anther is bilobed, dichoecous and tetrasporangiate. Pollen grains develop inside the microsporangia, 4 wall layers, the epidermis, endothecium, middle layers and the tapetum surround the microsporangium. Cells of the sporogenous tissue lying in the center of the microsporangium undergo meiosis microsporogenesis to form tetrads of microspores. Individual microspores mature into pollen grains. Pollen grains represents the male gametophytic generation. The pollen grains have a two-layered wall, the outer exene and inner entine. The exene is made up of sporopollenin and has germ pores. Pollen grains may have two cells, a vegetative cell and generative cell, or three cells, a vegetative cell and two male gametes, at the time of shedding. The pistil has three parts: the stigma, style and the ovary. Ovules are present in the ovary. The ovules have a stalk called funicle, protective integuments, and an opening called micropyle. The central tissue is the nacellus in which the archasporium differentiates. A cell of the archasporium, the megaspore mother cell, divides meiotically and one of the megaspores forms the embryo sac, the female gametophyte. The mature embryo sac is 7-celled and 8-nucleate. At the micropylar end is the egg apparatus consisting of 2 synergids and an egg cell. At the chalazoid end are 3 antipodals. At the center is a large central cell with 2 polar nuclei. Pollination is the mechanism to transfer pollen grains from the anther to the stigma. Pollinating agents are either abiotic, wind and water, or biotic, animals. Pollen-pistil interaction involves all events from the landing of pollen grains on the stigma until the pollen tube enters the embryo sac, when the pollen is compatible, or pollen inhibition, when the pollen is incompatible. Following compatible pollination, pollen grain germinates on the stigma and the resulting pollen tube grow through the style, enter the ovules and finally discharges 2 male gametes in one of the synergids. Angiosperms exhibit double fertilization because 2 fusion events occur in each embryo sac, namely syngamy and triple fusion. The products of these fusions are the diploid zygote and the triploid primary endosperm nucleus. In the primary endosperm cell, zygote develops into the embryo and the primary endosperm cell forms the endosperm tissue. Formation of endosperm always precedes development of the embryo. The developing embryo passes through different stages such as the proembryo, globular and heart-shaped stages before maturation. Mature dicotyledonous embryo has 2 cotyledons and an embryonal axis with epicotyl and hypocotyl. Embryos of monocotyledons have a single cotyledon. After fertilization, ovary develops into fruit and ovules develop into seeds. A phenomenon called apomixis is found in some angiosperms, particularly in grasses. It results in the formation of seeds without fertilization. Apomix have several advantages in horticulture and agriculture. A phenomenon called apomixis is found in some angiosperms, particularly in grasses. It results in the formation of seeds without fertilization. Apomix have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seed. This phenomenon is called polyembryony.